Hello everyone, my name is André Carvalho. I work at IPMA, Portuguese Institute for the Sea and Atmosphere, and I will present a study on Tanaidaceans from southern Portugal, an overview with new species and first records of Apseudopsis. This study is a result of a multidisciplinary collaboration between several Portuguese and French partners and colleagues. Tanaidaceans are a group of pericoid crustaceans widely distributed and abundant in sedimentary and crevical habitats. They are frequently found in large densities on shallow waters, accounting for a significant fraction of soft-bottom benthic communities, and they are an important food source for many trophic levels. Overall, 25 species of genus Apseudopsis have been described worldwide. 17 occur in temperate northern Atlantic, and 10 are circumscribed to the Mediterranean Sea. However, the real number of Tanaidacean species is clearly underestimated. Due to a high intraspecific variability, Tanaidacean species are often misidentified, especially in the occurrence of sympatric species of the same genus. In Portugal, specimens of Apseudopsis were recurrently identified as Apseudopsis latreillii. Such standard identification was presumably due to the lack of updated and specific literature or the invariable use of the same identification keys. Aware of the misidentification issue, we decided to develop this subject. This study aims to use a thorough integrative taxonomical approach combining morphology with molecular analysis to identify and describe putative new Apseudopsis species and compare different specimens from southern coast of Portugal and France. The study analyzed biological material from different sampling surveys of macrobenthic communities, from the Algarve coast and Rio Formosa lagoon in southern coast of Portugal, and from Bay of Banoules-sur-Mer, southern coast of France. We adopted morphological terminology documented in specialized bibliography. DNA was extracted, amplified, and sequenced. To search for matching sequences, we use all barcode records of family Apseudidae available on BOLD and GeneBank. Initial work resulted in describing the new species Apseudopsis formosus from southern Portugal in 2019. Apseudopsis formosus differentiate from congeneric Tanaidaceans, being unique within the genus in having the presence of combined morphological characters, especially short pointed rostrum, the 5 spines on propods of preoped 1, and mid-dorsal triangular wing-shaped protuberance on male Merus calliped. Besides the new species Apseudopsis formosus, 5 Apseudopsis species were recorded for the first time in the Portuguese coast. 4 were reported for the first time out of the Mediterranean Sea, and 2 outside the location of their original description. It is unknown if this distribution corresponds to a recent range expansion or if these species have been misidentified and therefore overlooked. Indeed, these records highlight the diversity of this genus in southern Portugal and reveal geographical connections within the Northeast Atlantic and Mediterranean Sea. Recently, we verified that some specimens found in France were morphologically similar to others found in Portugal and different from all described species to date. For this reason, we started a genetic analysis study, which is still ongoing, and the presented results are still preliminary. As you can see in the maximum likelihood tree, both specimens from France are together and have the same sequences, 100% of similarity. Morphologically, these specimens were identified as a putative new species, so potentially have a new species for science to be described. The results show that our sequences are regrouped in the same clade with other species of same genus, proving that our sequences are closer to them. Although the nucleotide sequences of present study specimens are really different, clearly indicating a different species. The present study provides valuable baseline data for future works in southern Portugal, highlighting the relevance of biodiversity in transitional ecosystems through the description of a new species and wider report of 5 first records. In addition, this study prompts the revision and updating of the genus Apseudopsis, improving the current knowledge on this abundant and ecologically relevant group. Actually, and to avoid misidentification problems, additional caution is needed in the taxonomic identification of cosmopolitan Apseudopsis species. Additionally, this study may contribute to improve DNA online databases, increasing the available sequences for Apseudopsis. This also highlights the importance of the integrative taxonomical approach, combining morphology with molecular analysis to identify and describe putative new species and new occurrence. This study was performed within the framework and financial support of the projects SNBSU4 and Marbis and MARI. Thank you very much for your attention.